today I have the honor to welcome Maria Mota as a speaker today. So Maria Mota did her PhD in the University College of London, moved to a postdoc in New York University, and in 2002 moved here to IGC to start a group as a group leader. She moved then in 2005 to Instituto de Medicina Molecular, IMM in Lisbon, where she's been a group leader since then, and since 2014 that Maria is the director of IMM. Throughout all this time, she's been interested in malaria, in Plasmodium. When she describes her career, many times sounds like it was a call to do science for the good of mankind, and to do that through studying actually a very important human disease. And she's been interested in looking at the different stages of Plasmodium in the infection, with a focus on the liver infection of Plasmodium, seeing how the host immune system reacts to the Plasmodium and controls or not the infection, and how nutrition and metabolism of the host affects the infection. Throughout her career, she received many scientific prizes, but also some non-scientific prizes, societal prizes. And I just want to highlight that Maria is a commander of the Order of Prince Henry, which is a high honor from Portugal. And she's also received the Prêmio Pessoa. And both these prizes reflect how Maria has also an important role as a face of science, as communicating science to the Portuguese society. And now I leave the stage. Thank you.
Thank you, Luis. Thank you very much for the introduction. Thank you very much for coming to this seminar. It's a pleasure for me to be here and do I need to do anything? Yes.
Yes.
I know. Sorry. Okay. Thank you. And it's a pleasure for me to be here. As we said, I spent 3 years of my life where I, in fact, started my independent career. At that time, Miguel Soares was already here. So, in fact, I will mention him a little bit later as well. So it's always a pleasure to come back. So I decided for this talk here, in fact, a little bit to honor the people that through the pandemic, okay, and this is the people in my lab, that really work very hard. They always work very hard. We all work very hard. But during pandemic was, harder to really be there, be more isolated, everything. So I decided to focus on projects that were really developed and finished during the pandemic. And this was just our lab retreat that happened on March 2021. And if you remember, this seems to be centuries ago. It was really a bad time, okay? Really, we have been coming out of a wave, and still we have found ways of being together and have a retreat and discuss amazing science. And the reason why I even decided to do this is because we were 14 at that time. And from these 14, 7 are not there anymore. So they left really during that year. So it's to these ones that, I think we should really talk about what they have been doing. And basically, this is what fascinates me. So what you are seeing here is the liver section of a mouse that has been infected with Plasmodium parasites. And basically, when the parasite reaches one of these cells here, each of these is like one cell in the liver. The vast majority of this parenchyma is made by hepatocytes, and the parasite infects these hepatocytes, and the parasite is tiny compared to the host cell. But very quickly, and in the mouse takes approximately 2 days, this parasite multiplies so much that the cell needs to increase 40 times in volume to be capable of really surrounding the entire parasite. And we are really fascinated by this process. What is so special about these cells that allow this enormous parasite to replicate there? This is just one parasite that gives rise to thousands of new parasites inside this cell. And we know that this proliferation, the proliferation of this parasite, the parasite is called Plasmodium, is the causative agent of malaria. Sorry, I didn't do any introduction because I even thought that was for the net. So, but this is, we are talking about a parasite that is called Plasmodium and is the causative agent of malaria. The Plasmodium is a parasite because it really lives, depending on the hosts. In the case of Plasmodium, depends on two hosts, the mammalian host, so any other vertebrate, in fact, and you will see here, as well as a mosquito that is not only the vector, but in fact is the main host. Okay, is the one where the parasite has sexual reproduction. Yes, this is a parasite. It's called parasite also because it's an eukaryotic organism and already has sexual reproduction, etc. So, but the thing that you have seen there is this process here, is the parasite that coming from the salivary glands of the mosquito, that when transmitted to mammalian, any mammalian, in that case that you saw there was a mouse and a human basically goes to the liver, and each parasite really gets surrounded by a membrane that we call parasitophorous vacuum membrane, and I will mention this, and replicates into 10,000 up to 50,000 new parasites, each parasite. And this is unique because even if you have the same stage of the parasite and some of them get stuck in the skin, yes, they may reproduce or replicate into 50 different parasites. Later on, at different stage, reach red blood cells and they multiply maybe into 10, 30 parasites. And so the liver and the hepatocytes in the liver have really something special that allows the parasite to reach this replication. If we go to another vertebrate, so in evolution terms, there is a lot of different Plasmodium species that infect any kind of vertebrates, including fish. Okay, so if you go to all these other vertebrates that are not mammalians, they also have a pre-erythrocytic phase, which means that before they reach the erythrocytes, they also have a pre-erythrocytic, but they don't go to the liver. And they go to some cells in skin. But again, there, they produce probably between 50 to 80 different parasites. So it's really something that there is very, very special about this liver environment. And this has been the question of our team for the last 20 years. So just for you also to have a better idea how it looks like, this is exactly what you have seen in the other section. This is a microscopy section, so then is being through the computer, and every single color here is DNA. So you see all the nucleus of the different cells of the liver parenchyma, and then you see the parasite there. The only understanding that you have is this membrane that surrounds the parasite, and we can now quantify in every single parasite, we can quantify these nuclei. So we know exactly that some parasites make 8,000, some parasites make 15,000, some parasites make 49,000. We can quantify completely all this. So when we have these observations, we always, and as I mentioned, the main question that we have been having is why are liver and hepatocytes so special? And as I also mentioned before, we've been asking this for the last 20 years, and we still don't have the answer. I should say that now we think we may have the answer because of some results that we have been having in the last few months. But if you would ask me 5 years ago, I would say, I think we are already there, okay? So we have not stopped. This question is really hunting us, okay? And of course, during the time, we discover other things, but we have not found the answer still to the main question, that is what makes hepatocytes so special. I'm going to mention some projects that are within this big umbrella, under this big umbrella of this big question. And one thing that is very special about mammalian livers is the fact that there is special heterogeneity. Basically, you have the portal vein, there's central vein, you also have portal artery, and each of these hepatocytes are bounded by this blood circulation, and of course, levels of glucose, levels of oxygen, etc., have been known for decades that they are different depending on these regions. And because of this difference, the liver is divided in what is called microlobules that have really functional differences within these cells. The laboratory of Shalev Iskarovitz and Weizmann Institute has shown in 2017 a beautiful paper that they show that by single cell transcriptomics, they can now have like a kind of a GPS and localize these hepatocytes exactly in the position of these microlobules. When we saw this, we thought, okay, so this is really a perfect tool for us now to ask two questions that we had at that time. One, it was, so when the parasite reached the liver, does the parasite choose the type of hepatocyte where it ends up? And yes or no? And if it's really, even if it's no, what happens to the parasite in different hepatocytes? We know that, but now the quantification that I show you with the nucleus, we know that some parasites replicate into 50,000 and replicates in 8,000. So what exactly is the difference there? So we contact Shalev, or in fact, we contacted here at the Fundação Carlos Gulbenkian because he came here to give a talk, not at IGC, but at Fundação Carlos Gulbenkian. We spoke to him and we start to work together. And so all this project was made really during the pandemic. Basically, the concept here, it was, okay, let's have mosquitoes, take the parasites from the mosquitoes, infect mice, and do time after infection through several time points. We made 4 different time points and have these questions. What is happening to the parasites? Are really randomly distributed in the liver, in the beginning, in the end? What exactly is going on there? So we have learned a lot of different aspects, but was one particular aspect that we learned that was quite unique. We found that around the time of 30 to 36 hours after infection, we could observe something that we call abortive cells. What are these? Are hepatocytes that have parasites inside that have a completely different transcriptomic type of response and really look like, you know, for all this type of response, our sense is that they were going out of the normal development and replication. In fact, we also did by in situ imaging, we really could detect this type of infected cells. And in fact, we showed that these parasites were basically dying and were not able to fully replicate. They show a unique transcript change. And one interesting thing is that they were all periportal zonated. So in fact, these all studies show 2 things. First of all, when the parasite comes out of the salivary glands of the mosquitoes and reaches the mammalian liver, it really goes wherever it can. So it's totally random, the location of this parasite initially in infection. But then when it really starts to replicate, is not random, the success of this of the infection of the parasite in the liver. Okay. And so this was something we know that this is just the tip of the iceberg at the moment. We really are now doing a lot, much more studies to understand exactly what is going on. But basically, it's what you can see here. You can see here a parasite that is really developing well, and a parasite there that is not replicating and is going through the way of being abortive. And that depends on the location and the type of hepatocyte that it is there. So one of the other questions that we have under this big umbrella of what is so special about liver and hepatocytes is also on the interface. And we are here on a kind of a networking of microbe host interactions. And basically one thing that we are very much interested is in the interface that exists between the parasite and the host cell. So what you are seeing here again is one of the, like the first image that you saw. This is an hepatocyte. This is the nucleus of that hepatocyte has been infected by a parasite. This is around probably or a parasite after 24, 36, probably around 30 hours of parasite replication there. And the parasite is surrounded by this membrane. It's not the plasma membrane of the host cell. Initially, it is an invagination of the plasma membrane of the cell, but then the parasite resides in this vacuole. And you have heard probably from toxoplasma is the same, how many of these parasites resides in these vacuoles. But we are very much interested how this membrane, as it is in the interface, how important it is for this. If you read the reviews and throughout decades, it says that must be important for host cell parasite communication, for parasite protection, to feed the parasite, for nutrient transition, to modify the cell, etc. But really, until very recently, we didn't have any single molecule that was present in parasitophorus vacuole that we knew its function. We knew that there was a lot of parasite proteins in this vacuole, that the parasite basically, places them and become resident of the parasitophorus vacuole membrane, but we didn't know the function. And I'm not going through all these studies that were done through the lab, but we started basically back in 2015 to work in some of these molecules, and we could show that some of these molecules interact with all-cell autophagy machinery to block it. Now, more recently, VDR has really showed that one of these molecules interacts and manipulates the whole cell actin to protect the parasite from being extruded from the whole cell. And we also sometimes have surprise, and João Melo Vieira was a student that really showed that a molecule that is in the vacuole of the parasite, but in fact is so important in the formation initially of the parasite, that parasite does not even reach full invasion when it is deficient of this molecule. But the work that I'm just going to highlight is the work of Aparajita Lari, and Aparajita was a PhD student in the lab, and basically what she has done, it was a screen for a huge number of molecules of the host, because until now we were really focusing on molecules of the parasite that reside in this parasitophorus vacuole membrane. But of course there are molecules from the host that are there, and we wanted to know which ones were there and what was their functions. And she did a huge screen. This is not still published. We are still working on all these, but many of these, basically she was looking for all the type of endosomal type of signatures that could be around the PVM. And she found that with module liver stage, they really selectively divert distinct host cell compartments. There are some host cell compartments that are really avoided from there and others that are in there. And so with this, she really focused on one particular molecule, and this is APPL1. APPL1 is really an adapted protein that is present in very early endosomes. And basically what she showed is that it was there. She showed the localization of these, and basically, and very important, she constructs tools that allow either to reduce the amount of this protein in the parasitophorus vacuole membrane, or increase its amount in the plasma membrane just by, reducing in the host or really sequestering in the host. And when she did this, she showed that this protein is, the parasite basically sequesters this protein in the parasitophorus vacuole membrane to allow not function. So the parasite doesn't hijack any function of APPL1, just it sequesters to really grab it and make sure that this protein doesn't make its own function, and the parasite can really progress if it does that. If it's not able to do that efficiently, the parasite can replicate much less. But Louis shared that we are focused on the liver stage of infection, and it's true. The vast majority of our lab works on the, this stage where the parasite basically is transmitted by a mosquito and replicates into this huge number of parasites within hepatocytes. But the truth is that, this is not even malaria. So it's not even is an infection, but it's not a disease. The disease only starts when the parasites are then released from the liver and infect red blood cells and go through cycles there, okay? And we have been interested for a long time. And the first project that we did about this was with Miguel Soares here in this institute, that have been very much interested why some people die, why some people don't die with malaria. The truth is that we have more than 300 million new infections every year, and we have around 600,000 deaths. Of course, 600,000 deaths is terrible because it's children below 5 years of age. So it's a huge number and it's an acceptable number, but compared to the number of infections that we have in the world, it's really a small proportion. So what makes people to die or not? And we were not new, Miguel and I, to ask that question. That has been a question that has been on the minds of researchers for more than 100 years ago, and many things were discovered, like sickle cell trait and all many aspects, okay? But one aspect that, and we became interested more recently, is in fact something that was very different from what we did with Miguel Soares, is that there is a kind of correlation in the amount of parasite load in the organism and the disease severity, okay? So Miguel will defend that independent of the parasite load, okay, you can have that or not. And yes, it's true, totally. But if you look in the field to a kind of a study, and there are many studies, in fact, if you measure total parasite load, basically you can see that there is a correlation with severe malaria and higher parasite load. And so, how is this parasite load? What is the result of what? And can be the result of two things. One, a parasite that replicates more. The other, an immune system that controls more. Of course, disease and etc., as I'm sure you have heard from Miguel Soares, will depend on many other aspects. But in fact, parasite load itself depends on the capacity of host to clear or on the capacity of parasite to replicate. And in both cases, depends a lot on the type of parasite and host. And this has been really a dogma there that, you know, it depends on the genetic background of the parasite, because some parasite lines really and some parasite strains really replicate more than others, and depends on the capacity of our immune system to control it, okay? But in fact, some years ago, and this was before the pandemic, but a postdoc in the lab, Liliana Masso Silva, that is now a group leader at Pasteur Institute, basically Liliana showed that there was another way, in fact, of parasites replicating more or less. And that was totally independent of the genetic background of the parasite and was totally independent of the host genetic background. So you can have exactly the same parasite, exactly with the same host, but if the environment contains high levels of nutrients, the parasite replicates more than if it contains lower amount of nutrients. As you can see here, basically here you have two mice, and you can immediately see the one that is in conditions where it's more than the other. This is in ad libitum conditions. The mouse, like all animals in our animal houses, they eat as much as they want whenever they want, and is why it's called ad libitum. And then this mouse has been placed for 2 weeks in a system of caloric restriction. This is not undernutrition. The parasite has only 30% of the calories reduced. As you can see, is already more elegant, okay? After 2 weeks, 2 weeks later, you infect with a parasite that expresses luciferase in this case, so we can immediately detect the entire parasite load in the entire body. And as you can see, 2 days later, after being infected with the parasite, now on blood stage, and this we are talking about blood stage, and is why it's all over the body, basically the one in caloric restriction have less parasites. After a long time of Liliana looking what was going on, she realized that the parasites under an environment of low calorie, the parasite every time in red blood cell replicates in lower number of parasites than the one in ad libitum conditions. Of course, at that time, you could imagine that maybe this is just a passive type of response. Basically, you have a parasite, there is low nutrients available, maybe there is one nutrient that parasite needs more, and parasite is struggling to make more number of replicating parasites, so cannot do it more. Or you could hypothesize that in fact that is not the case. It's an active decision of the parasite under certain environments that will decide to really immediately replicate less. And we know that the second hypothesis is one that is true. How we know that? Because Liliana was able to find one parasite that is deficient in a kinase that independently of the conditions of the environment always replicate at high levels. So there is nothing that is missing because this parasite really can make the same number of replicating parasites in ad libitum or caloric restriction. It's just that now doesn't have a kinase that is able to sense the environment. And so this kinase is a sensor, and was also, again, a kind of a different shift in the way we're thinking, because although all our cells have systems, and in fact, you know, very canonical systems that are in terms of evolution present in many organisms, including in other parasites like Toxoplasma, and I have a system like, for example, AMPK, that is a sensor for energy. Plasmodium doesn't have any AMPK-like type in its genome, but there's one kinase that is called KIN. We didn't give this name, okay? This is just called KIN because it was the first kinase in the 80s to be cloned, okay, and this is why it's called kin, and nobody knew what is its function, and its function is a very simple but very important one, the ability to sense the environment and really allow the parasite to respond before struggling in certain conditions to respond and adapt to the environment that is around. Recently, and this brings me to what has been done during the pandemic, and this is a student, Inês Marreiros, and basically Inês Marreiros was interested that not only to look to the entire caloric restriction, but now to look to certain specific nutrients. And the ones that she decided to look, it was to amino acids. Again, why? Because this parasite is really a very strange parasite, also doesn't have a TOR or anything like TOR that could be used as a sensor for amino acids. And so it has been published more than a decade ago that the parasite, because it doesn't have this system, again, is not able to respond to changes in environment in terms of amino acids. And the only way that has to do is that if you remove one amino acid, the parasite becomes dormant until either it dies or soon enough the amino acids placed in culture, and this was all made in culture, and the parasite wake up and starts to replicate again. So Inês Marreiros started to play around with two different amino acids, isoleucine and methionine, and she showed that in fact what was published in culture was not what was really happening in an in vivo situation. The parasite was indeed able to respond, and again, by producing lower number of replicating parasites, as you see here, and here you just see, instead of a fluorescent picture, you just see in color, but basically she could show that they respond with making lower number of replicating parasites, but also by delaying, especially with isoleucine. So it's not exactly the same with all the amino acids, but especially with isoleucine, they have a delay in development and then they produce slower. But the interesting thing is that Inês was able to really make it completely independent, the two phenotypes. Basically, what she shows is that there is two kinases that are involved in protein translation that are active in different points of the life cycle of the parasite inside the red blood cells that really signal either to become slower or either to make lower number of merozoites. And by blocking one or blocking the other, you just block one of these phenotypes and they are not connected. So they really, you can have them independently, and this time, depending on in which part of the replication cycle, cell cycle, the kinase is involved and becomes activated. Then this brings me to, of course.
So we were playing with this idea that if one messes, as you've done or as we did, with replication of the parasite in a manner that the absolute burden is less under some conditions, we found some evidence that the parasite really begins to express genes that are involved in transmission. So I do not know. So under your conditions, do you find such a thing? Because within the context of a successful pathogen, you know, so now I'm trying to anthropomorphize a parasite. There should be a way that they will get out, or they should be able to continue a transmission. Otherwise, if I'm reducing my proliferation, what's the benefit in the end for me?
So the question here is when the parasite, so let me show you the life cycle. You have the mosquito, the parasite reaches the liver, multiplies like crazy, goes out into the blood, and now every 24, 48, 72 hours, the parasite goes through a replication cycle of few numbers of parasites there, okay? But what is mentioned here is that also for the parasites to be able to transmit to the mosquito, they also need to have what is the preparation for the sexual reproduction. And so the parasite, some parasites, when they infect red blood cells, instead of going in this, cycle over and over again, they go out of the cycle and they make female and male gametocytes. And these ones that when then a mosquito comes and bites you, they remove these ones and they make a zygote, an egg, inside of the mosquito, replicates like crazy, and then have parasites in salivary glands, okay? what you are asking is that, if you have a conditions that they replicate less, your goal is that, let me go out of here. If it's amino acids, yes. If it's energy, is not. My short answer is, so obviously in the beginning we thought, wow, this is going to be the thing, is that the parasite makes less parasites and really replicates and really goes out of the cycle to make sure it transmits as quickly as possible. We don't see evidence with that with caloric restriction when it's associated with energy. But with amino acids, yes, we see. As soon as we start to remove one of these amino acids, then the parasite moves out to the next part of the cycle. Glucose or fatty acids. So really, it's very difficult to play around on this because the parasite can shift very quickly from glucose to other sources of energy if we play around.
Did you ever try energy and temperature at the same time?
We never tried temperature.
Because so he's being shy about it, but he has shown that in vivo, they seem to respond
to temperature,
to energy
and
temperature. And 1Q is not.
I'm not surprised. I think you are going to find a huge amount of sensors in the parasite, as all other organisms have. The only surprise is that basically they don't have any of the canonical ones that exist from plants to yeast to mammalians, and Plasmodium somehow came out. Toxoplasma has some of these canonical systems and is the cousin of Plasmodium, and Plasmodium doesn't seem to
Yeah, I
have.
don't want to take too much time, but this is reminiscent of, for example, in the immune system, these two signal systems. So one is not enough to take a decision. You need one plus another one, then you, and it's a big decision to go into.
So
a
one project that I'm not showing here, obviously coming out of this project here in particular, we are looking for what are the effectors. What we found the sensor, but must be an effector for this response. And of course, the most logical ones to look for would be transcription factors. Plasmodium also is very strange in transcription factors because it has this family. So for many years, if you go back to 15 years ago, and I was still young, people would say Plasmodium doesn't have transcription factors. There was a time that even people thought that because they could not find the typical ones. Then they found this family of transcription factors that are reminiscence of transcription factors in plants, and is from this AP2 family, okay? And we basically took all this family, have all these knockouts, parasite lines that are deficient for this, that are not essential for blood stages, and play around. All of them are involved. All of them. We cannot even, we have now a person studying with us with mathematical models, see how this could make sense. All of them are involved. And I think they really come together and put together so many type of, things in environment. Basically, this is it, okay? So, but it's going to be very difficult to really take one apart of the other, okay?
So under, let's say, energy restriction, right? So the animal begins to use stuff like amino lipolysis from fat. So I'm
they also have less amino acids. They have everything reduced. Yeah.
So the amino acids that modulates the growth of the parasite, do you see them in this caloric restricted mice if they are more or less?
Yeah. All in this caloric restriction serum, we did the metabolomics of all this. Yeah. And basically you have all amino acids are reduced. Some of them only like 15%, some of them up to 50%, okay? And is very interesting on this, is extremely heterogeneous even among C57 black 6 mice. So if you do 10 mice, yes, you have a tendency to go more on one, but it's really heterogeneous. And the diet is all the same, the time of the diet is all the same. In the beginning, when we did the first experiment, we were doing, wherever was the person putting the diet, then everything is fixed, day of the time of the day, the amount of calories, everything is measured and still is heterogeneous. Okay. Anyway, so as I was saying, also another question that we have within the blood stage that the stage that causes disease is not only who dies or who doesn't die, but who dies and with what, okay? So malaria itself is not one single disease. It's really a group of different syndromes that in the end could be just multiple organ failure even, okay? And be something sometimes like even similar to sepsis, okay? And, you know, I'm sure that you all have heard here much more than I could say, but, you know, in humans itself, there is at least 3 syndromes. They are not exclusive. Sometimes they come together, but they are not randomly distributed. So we know that is not something that happens randomly. And you have either a neurological syndrome, severe anemia, but also you have respiratory distress that as the most common ones. But as I said, many times it's just multiple organ failure involved. We know for a long time, okay, that TC is both host and parasite, you know, dependent. And this doesn't need even, we didn't need to do any experiments whatsoever, but since the 70s, that we know that one strain of Plasmodium bergheianca in black 6 mice, all the mice, 100% of the mice die with a neurological syndrome, okay? But we also know that the same parasite, Plasmodium bergheianca, in a DBA mouse, they die with acute respiratory distress syndrome. We discovered this in 2009, you know, by chance, I should say. But they die with the same strain of the parasite, but it's just in a different strain of mouse they die this. But we also already knew that black 6 mouse now with a different strain of Plasmodium berghei that is 99% identical, now they die with the respiratory distress syndrome as well. So we know that depends both on the host and on the parasite. We knew, and from a long time, and this was the first study that was done and was done in this house, okay, and is with Miguel Soares, we show that there are host genes that in fact are very important for the protective response against this severe disease in case of neurological syndrome. And Miguel continues to show this in many other aspects of severe disease in malaria. But we also show that there were some host factors that were really detrimental and really were on the basis, for example, of acute respiratory distress syndrome. We did this in collaboration with Sergio Dias and was in VGF. But more recently, okay, and also in collaboration with some people here at IGC, Isabel Gordo and Ricardo Ramiro, we really basically looked to a different aspect. So we found this by chance. We were going to a completely different project, but realized that the lungs of the mice that really going to develop acute respiratory distress syndrome had a kind of a dysbiosis of the lung microbiota. All these mice present dysbiosis of the gut microbiota independent with which strain they are infected. But the mice that really develop, and this is both mice, black 6 mice infected with K173 that develop acute respiratory distress syndrome, or one that develop cerebral malaria, the neurological syndrome. Basically, what you see here is that in this one that develop acute respiratory distress syndrome is a loss of diversity of the lung microbiota. And I am not showing here, but the paper has been published already. We show a huge increase in the bacteria on these lungs. The microbiota in amount, it increases, but the diversity decreases, and is a typical definition of this biosis. As you know, you can see the difference here in this graph. And I don't really have the time to go into this, but basically what we could show is that in these models that develop acute respiratory distress syndrome, the main feature that we found the main difference that we found, it was that the infected red blood cells, which have a capacity to bind to the endothelial cells in a process that is called sequestration, okay, basically they choose the endothelial cells of the lungs mainly, and this leads to a host response and inflammatory response that the host tends to counteract with IL-10. But when it does IL-10, certain type of bacteria seem to be in an environment that they seem to be more prone to replicate, to grow, and they cause this loss of diversity, but increase in the number of bacteria. We show that if we really block IL-10 or the cells that produce IL-10, basically we could block the disease itself. We show that if we block the sequestration of parasite endothelial cells, the same. And we show that if we have antibiotic treatment, we could really transform a not healthy lung into a healthy lung. So in both these projects, we go back to the basis of the, of microbiology, that is the epidemiological triad. We have the host that we always like so much, and we have been always working on it. We have the parasite itself, but we also have the environment, either if it's nutrients, if it's the microbiota, that really is a very, very important component of the disease and the establishment of pathology. But now I'm going to finish, and I'm going to concentrate in the story that, you know, it was one of the most recent ones that we are very interested working on, and mixes everything that you have heard of. So basically, when we study, and all the projects that I showed to you in blood stage infection, basically what we use mice that are infected with infected red blood cells. Basically, we make a blood transfusion of infected blood from one mouse to the other. We quantify them. We can make them from 10,000 to 1 million infected red blood cells. We make these and all the models around the world for all different labs that were mouse models. They are like that, okay? But there was a question that we always ask, is that, does the liver stage, because, you know, we do this, but the truth is in natural infections, you have this going on in a body before the parasites reach the blood. So does the liver stage predispose the type of response that you have in blood stage or not? The problem is that we had this question, but it's very difficult to have a system that is comparable. Because we can have this by blood transfusion, but now if we have trying to compare with ones that we infect sporozoites, it's very difficult for us to know exactly at which day and how many parasites reach the blood. So it was really a different question to make. But we were really prone to do this because some years ago, we showed that indeed the liver stage have a huge response sometimes against some of the infected hepatocytes, meaning that, you know, the host has seen the parasite and even attacks some of the parasites. So obviously there was something that this parasite has been seen before going into the blood. So there was already some kind of clues that, yeah, the parasite will be predisposed to really, or the host have been seen already the parasite, and so would respond differently if the parasite has gone through the liver. So Angel Shora, and in fact he was a PhD student here with Miguel Soares many years ago, basically what he has done, it was trying to find a model that he could compare the two. And he really found a model that you would have parasites coming out of the salivary glands of the mosquitoes, going to the liver, but will be a non-productive one. So basically we can use irradiated parasites, but we can also use now genetically modified parasites that they make all the entire liver stage, but then they are blocked at the last step. And by doing this, then when you can do it equal numbers of infected red blood cells 2 days after the period of liver stage infection. So we have then used this model and developed this model and ask what is the crosstalk between these development stages. And here, this is going to be complex, okay, so, but stay with me. So if you have, and I use blood stage low just for 10 to the 4, blood stage high just 1 million of infected blood cells. If you have these injected into a mouse, okay, basically all mice die. This is black 6 mice infected with Plasmodium bergianca, so they die with a neurological syndrome, and they die with a couple of days delay because it's just justified by the number of parasites that the infection is started with, okay? You see the parasitemia, that is the percentage of infected red blood cells, is also, but they die more or less at the same level of parasitemia, just a 2 days delay. But now, if you have these same exactly mice that 2 days before they were pre-exposed to the liver stage infection, basically you have the liver stage blood stage high all die, the same as that, but the liver stage blood stage low, they all survive, okay? There is no difference in parasite load. This could not be justified from that. So there was something else going on. This is not only true for the neurological syndrome. If you have now, as I mentioned to you, the black 6 mice with Plasmodium bergianca K173, they die with acute respiratory distress syndrome, and you observe exactly the same. If you go just with blood, they all die. But now, if you go with this, the liver stage, blood stage high, they die, or the vast majority die, the other ones survive. So I'm going to tell you a very long story of several years, of a couple of years, on looking what could exactly be. And we end up in gamma delta T cells, okay? So basically what we show, and this was really surprising for us and very difficult to understand for some time, is that if we use mice that are deficient in gamma delta T cells, now the liver stage, blood stage low The wild type all survive. But now, if they are deficient for gamma delta T cells, they start to succumb to the disease. However, in opposite now, if you have liver stage, blood stage high, the wild type, the vast majority succumb to disease. But if they are deficient to the gamma delta T cells, basically they start to survive. How come the same cells have the opposite role and the opposite phenotype? And I can tell you that, well, for us was months of really like knocking our head in the wall, exactly what was going on. Of course, we were very lucky because next to us is the laboratory, the lab of Bruno Silva Santos, and they are experts in gamma delta T cells, and we start to discuss with them. And I thought, okay, guys, maybe it's just two different types of gamma delta T cells. And so gamma delta T cells are many different kinds, okay? But they have the tendency to exclusively produce either interferon gamma or IL-17. So they ask what is happening in the livers of your mice. And basically what we could see, so basically you have on the left side the liver stage, blood stage low, the ones that survive. You have on the right the liver stage, blood stage high, the ones that will succumb to disease. On the top you have interferon gamma, on the bottom you have IL-17. As you can see, the ones that survive, basically they have very low levels of interferon gamma, but they have a huge number, or an increase on the number of cells that produce, and I have one of these, sorry, I should use, okay? Now I realize, sorry about that. And that they produce IL-17. On the opposite, the ones that succumb to the disease, now they have high numbers of cells and increasing cells that produce interferon gamma, but very low number of IL-17. Of course, and this is just the ratio here. So of course, then we ask, so what happens? Can we remove one of the others? And there is a way. So there is some transgenic mice that basically, they produce all the gamma delta T cells except the ones that produce interferon gamma. And when we do this was very, very interesting. In the system that they usually survive, this mouse behaves as wild type. But in the system that usually the mice die or succumb to the disease, they behave as if they were gamma delta knockout mice. So what happens now if we take IL-17? So we don't have a way of taking IL-17 specifically off gamma delta T cells. So we use IL-17 deficient mice, but now again, the result was same. So now in a system that usually wild type survive, now the IL-17 deficient mice behave as gamma delta knockout mice. But if we are in a system that usually the mice succumb to disease, now they really don't behave as gamma delta knockout mice, they behave as if they were wild type mice. So this I know is not something immediately easy to understand, but is really you have the same cell that is triggered by liver stage, but somehow these cells have two types of cells or two types of families of cells that produce either interferon gamma or IL-17, and this really dictates if these mice are going to succumb or to be or to really be protected from disease. So, but what does gamma delta do? And again, we went through the entire body of the mouse. Obviously, we lose a lot of time in the liver, but went to other organs as well. And then we realized that very early of the infection, basically we have an accumulation of gamma delta T cells in the spleen of these mice, and especially in the mice of liver stage, blood stage low, but not in liver stage, blood stage high. So we wanted to understand the mechanism of protection, and I thought if this had anything to do with protection. To go into that, basically what we did it was to have mice, now wild type mice, and because these wild type mice, they are protected in this system, liver stage, blood stage low, but if we spleen optimize them, basically they lose all the protection. So the spleen is indeed important for this protection. So what is this spleen doing? And again, we look to the entire type of cells that this spleen was producing. And one thing that we observe is that compared to non-infected, the ones that were protected have a huge increase in erythropoiesis. Okay, and most important is in the mice that they are not protected anymore, either the gamma delta knockout or the IL-17 deficient mice, this erythropoiesis is lost. So then the next experiments that Angel did, it was very simple. Just transfuse reticulocytes from one mouse to these mice that were really not protected anymore, or make a transfusion of normal red blood cells. So the controls here, they also receive a transfusion, but normal red blood cells, while our experimental group receives a transfusion of young reticulocytes. And basically what we could see is that in both situations, either in mice that are gamma delta deficient mice, that basically, they also come to the disease. Now, if they are transfused with young reticulocytes, they start to survive, and same for IL-17 deficient mice. So basically, what we are proposing as a model is that when the parasites go through the liver, it really is a kind of a first signal, first trigger, and triggers and preconditions gamma delta T cells. Doesn't make them to go in one way or the other. They are just preconditioned. And then depending in time and space of the amount of antigen that they are exposed, and this is the difference between a fast high amount of antigen or a slow low amount of antigen, basically the gamma delta T cells then can functionally become distinct in either producing interferon gamma or IL-17, and this is correlated. And I don't show here, but this is correlated in this case with an increase of the sequestration that I showed to you, that is this binding of infected red blood cells to endothelial cells and causing damage and severe disease, while here leads to an increase in the production of reticulocytes, of young reticulocytes, an increase of reticulocytemia from the spleen that somehow decreases the sequestration that we observe, and is something that we are interested to understand. The paper is coming out tomorrow in Immunity, and Angelo is very happy the way that the pandemic turned out to him, okay? And so just to finish, to thank all the people involved and all the members of the lab. I think I mentioned all of them, and of course all sponsors, and thank you for your attention.
Yeah, I got lost, I'm happy to say.
Mm-hmm.
But still, at the end, you said something which was a bit surprising. You said it was you said something to do with the burden of antigen.
Mm-hmm.
Do you know it's antigen, or is it a cytokine responsive?
I mean, in this response here of the gamma delta, etc.
Yes.
Mm-hmm. We know that second trigger comes from here, and the only difference that we have between these is time of the amount of antigen. What else can we have?
Yeah, but it's not. I misunderstood maybe, because I thought you were saying antigen recognized by the gamma delta T cells. You're not saying that. No. Okay.
Not at all that. What we are saying is that the liver stage preconditions and somehow is the first trigger for gamma delta T cells. If you don't have that, and it's something that I don't show here, but if you have just the normal blood stage infections that we have constantly,
Yes.
You don't have any of this. So the liver stage really preconditions these cells to be alert. And then depending in the time and the amount of antigen, because it's the only difference that we have, basically these gamma delta T cells can go one way or the other.
Yeah.
But doesn't mean that they recognize anything on the specific antigen. I mean antigen in a way of amount of parasite, okay? Of trigger.
Yeah. If you'd use an alpha beta negative,
If I use
if you have an alpha beta negative mouse,
Mhm.
then does this phenomenon not happen? Or do you,
No, if we have that, is normal. It's like wild type.
it's like wild type.
I just want to ask a question about this. So in a normal infection,
Mhm,
you have this pre-stage of the liver,
mhm.
and then which of the two scenarios is the next stage of infection?
In the field.
Yes, the gamma delta T
Mhm.
cells are responding to a slow, low parasite mass increasing, or are they responding to
Depends.
a high mass release of?
For how long have you been living in endemic area? So in fact, we think this is part of the response of a natural immunity in living in endemic area. So we also have a mathematical model to show how exactly it happens in endemic area, where we basically have people are always exposed to infected mosquitoes. We have regions with 300 mosquito bites per year in high endemicity. And so the amount of parasites that can come to the blood can be much higher in the beginning when you don't, you are very young, etc., and then becomes slower, lower
So you
later
think
on.
then the gamma delta T cell response will change with
Will
each
change
exposition?
with the exposition, not the
Exposition, yes.
the exposition.
So in this dichotomy in the liver, do you see differences in the presence of neutrophils versus macrophages?
Mhm.
Because I would expect downstream of IL-17, perhaps you have more neutrophils, and with interferon gamma, you would have more active
But where? You
macrophages
mean
in the liver itself after.
Not, no,
No
you
differences.
don't see no difference in the liver. This doesn't happen. This you need to see that it's two triggers. You have the liver, and you don't see all these changes, for example, in spleen. You need to have the liver and the blood stage. It's two triggers. It's like kind of innate immunity for inflammasoma. You need the two triggers to reach this level here. Hmm?
I'm sorry. I didn't get exactly the, so the blood stage high and the blood stage low. I'll be more specific.
Mhm.
So when you give more parasites,
Mhm.
I'm assuming that like the total number of red blood cells is always the same,
Mhm.
or
Of course. Exactly. So the red blood cells is the same. It's just the parasitemia is higher.
the parasitemia is higher, but the total number is always the same.
Mhm.
Okay, so it cannot be that. So my theory is
You always need to control
Yeah,
for
yeah.
that, of course.
because it could be. And this is more.
Is why you also need to have a control, a transfusion of normal red blood cells, correct? Because if
Yeah,
not,
I'm digesting
could
it still.
change.
So yeah.
Mhm.
And for the gamma delta T cells, it's so Mark will, I always say his name wrong. Voldoen.
Mark Eldoen, yes.
Yeah, he is world known for AHR and IL-17.
Mhm.
But he did this on T cells. So my question is, gamma delta T cells,
Mhm.
do they also use, do you know if they use this receptor AHR to become IL-17?
I don't know.
I think,
That I don't know.
yeah, that would be really cool because what might differentiate them. So Mark discovered this for T cells.
Mhm, I know. Mhm.
Uh, what might differentiate?
I don't know, but might be known, okay? Because I don't know then enough from
Yeah,
this
yeah, I don't know.
gamma delta T cells,
Yeah,
okay? So
but you probably will know.
Mhm.
So that would be a good target to define this dichotomy. Why do they have interferon gamma or IL-17? Because if they have a ligand of AHR, and there's many during malaria,
Mhm, mhm.
this might dictate where they go.
I have a curiosity. In the beginning of your talk, you show these very big cells, the cells that become enlarged and enlarged and enlarged. Where does the membrane come from?
Fantastic question that we have for more than 20 years. We don't know. We did even a huge project with Andrei Shevchenko at Max Planck Institute to do a lipidomics to see what is the source. And it's really very, very interesting. So although you have a tiny cell there with a very tiny parasite, parasite grows, 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 grows. Toxoplasma is different because they grow in this number, they go to next, but this one grows, 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 and you would imagine that will be completely different in lipidomics. Yeah, a few differences, but nothing very spectacular. Nothing very, it seems like everything that is now in the host somehow, the equilibrium is maintained. Somehow this has evolved, as we can imagine, that to really be in this equilibrium in the end and infected hepatocyte and the non-infected hepatocyte, I can point to 4 different differences that are really, nothing very special. So we don't have any idea. No, not idea at all. And it's not only that. It's not only the where comes the membrane for the hepatocyte itself, but for the parasitophorus vacuum membrane and for doing every single parasite in the end that is only made in the end. So these are nuclei that are all in one cytoplasm and divide in a synchronous way. Then they have one last replication that is synchronous, and then they start to cellularize. Yeah. So it's very interesting because the majority of these organelles, the ER, the mitochondria, and another organelle that is parasites have that is called apicoplast. That was really, so basically they are all connected. So throughout all the replication is just one that grows in size and goes all around, and only is cut at the last moment.
Maria, I wonder about, I have two questions. The protective role of the gamma delta 17 producers in erythropoiesis, is this a common role that these cells have in extramedullary erythropoiesis?
Yeah, we were surprised, but in fact, there was already a paper. So even the people, we were surprised with everything because we didn't know anything about these cells, okay? But even the people, our collaborators on gamma delta T cells, they thought, how could be these erythropoiesis? But in fact, there was a paper, blood paper, that already shows that the gamma delta IL-17 produced cells lead to erythropoiesis. And this blood paper, I cannot say that is the strongest evidence ever, okay? But there was already a paper showing that. But it's true, now we have done IL-17 knockout, all this, and makes injuries to see in aspects that they can really respond, and they respond less. So is something that, yes, they have a function.
And the second question has to do with the dichotomy between the low and the high parasite load. I mean, do you have this protective level that is induced in both mice, and then when you have a high pathogen load, you just have this megalomanous expansion of the interferon producers, or do you know how exactly these differences are
So it's
operating?
not dependent on the load of the blood stage per se, because if we have just the blood stage independent of the load, they die exactly the same way. So it's not exactly that. The way that, and I'm not so sure if I'm answering you this way, but do at least all the data, and we have much more data than this because we have been back and forth trying to understand what is going on. But the way that we see it is that there is a kind of precondition by the liver stage, and they are prepared to go one way or the other, but we don't know exactly what makes one go one way.
That was my question, whether you have a major shift in something that they can still decide to be, or whether you just have expansion of the interferon gamma producers.
So if you would see that, then it would be the opposite, because when you have just blood stages, basically they seem to have just the interferon gamma response. That could be the thing, but it's not true, exactly what I'm saying. Why? Because then we went back, why all the blood stages always die, independent if it's low or high? They don't use gamma delta. So if you use gamma delta knockout mice, there is something else. Okay, so it's a different system. And now we know, even through the pathology, that they die with the neurological syndrome, but the path to go to the neurological syndrome is slightly different.
So, hi, thank you for the talk. Great. In your dysbiosis model, if you block dysbiosis, or if you remove the bacteria that over-proliferate, is the disease less severe?
Yes.
Or are the mice protected against?
So in every single cascade of that, if we stop at any step, then you don't have dysbiosis, and then you don't have severe disease. Severe disease depends on this dysbiosis, at least from at the level that we could prove. Okay.
Can I have a second one? From your previous story on APPL1, if I remember correctly, it interacts with the adenopoconnectin receptors, which regulate lipid uptake, glucose uptake, and so forth. So is there a role for that in the liver? Is the parasite regulating the?
Yeah. No, it's not regulating. The parasite makes sure that it does not do that function. So basically,
Yeah, but in that sense, is it the way that it's working?
yes. So we don't have the proper mechanism. So it's not that we really could find all the proper mechanism, but for a couple of years, we were really thinking the parasite uses the function of this now into the parasite. And because there's so important functions on this, then obviously the parasite is not. Is non-functional when it's in the parasitophorus vacuole membrane, and needs to be non-function for the parasite to fully replicate. So basically, it needs to stop its normal function in the early endosomes.
Yeah, I understand, but it would still be manipulating the levels of lipids, glucose, and insulin signaling within the hepatocyte, which might be relevant for the way that it replicates.
You will see that. And correlates with these levels when we have higher or lower. Okay.
Okay, we have a final question, because if not, we cannot leave.
Okay, with respect to nutritional demands, is the nutritional range in Africa, I mean, in hypermalarial areas, such that you see
Everyone asks me that.
Oh,
So I don't take it in a bad way, okay? So, because everyone asks me, okay, but now if in Africa people eat less, imagine that they start to eat more. So let's starve them. No, this is first is one mistake that we have. You need to imagine that at the moment, okay, almost 40% of people in Africa are already reaching obesity. So it's really becoming very fast. So this is not something that sometimes is a stereotype that we have, and is not exactly the case, okay? But we think these systems, these sensors, they work in alterations. It's like your AMPK. Your AMPK really senses changes. Is why you become insulin resistant. If you start to eat all the time, then your sensor is not working anymore, correct? So what we think it is that parasite detects alterations and changes, okay? I don't think it's going to be by playing the amount of nutrients that we would be able to resolve anything. But for us, what we thought in the beginning, and I think is not true, okay, but for a long time we thought it could be, we were very happy to think about this hypothesis, is that this could be, in terms of evolution, this evolved for people living in dry and raining season. Dry season, nutrient levels go really, really low in populations, okay? So it's a change. And there is no mosquitoes around, so could even explain why even in caloric restriction you don't have gametocytes, because there are no mosquitoes. So basically you need to go into energy saving pathway. You just go low, you just go, okay. Nowhere to go. And there is no, okay, I need to go low to make sure that I don't kill this host, because I need to wait for 6 months to come mosquitoes again, and so could be a signal. So one of our students, Silvia Portugal, she is now a group leader at Max Planck Institute, and basically she works now in the field with samples from the field. The problem, and she really worked very hard, looking for if really manipulating this could be explained in serum from people in dry season, raining season. She works in the perfect place in the world for this, because she works in Mali, that they have 6 months raining season, 6 months dry season. This would be the perfect for this. She cannot make it work, okay? She stopped to believe on it, okay? To me became quite almost a religion, so I cannot stop to believe on it, sorry. But it's because the problem is that these experiments are very difficult to make. Why? Because as soon as you take the serum and, because you don't grow parasites in serum, you grow parasites in a medium that in the majority of the times is so much more rich than anything that is in serum. So it's very difficult to manipulate. So I cannot tell you that we have any evidence that this is true, but I still like the idea. Just, okay.
Real world. Sorry, just I can ask you after. Okay, I'm sorry. So the work of Wilson and Susanna, we don't understand why that is, but, and correct me, Wilson, if I say wrong. So we forced hypoglycemia, which is one of the factors that you show in caloric restriction that is very important, right? So we force it genetically, we switch off the...
Of course, you need to think that caloric restriction never goes to level of hypoglycemia.
Yeah.
These mice are more healthy, okay?
Yeah. But
so
I'm just pointing out that the system is different. So we just
drop it down, really.
And the parasites, they change, but not all the way. They don't become gametocytes. It's only if we also change their temperature that then they go all the way. So I'm wondering in the dry season, right? So they might have some, well, traditionally or evolutionarily, some sort of caloric restriction, but the parasite doesn't get triggered by that because there's nowhere to go.
Exactly.
Unless there would be another change. In our case, we found temperature, but it would be something, and then it makes sense to keep fitness of the parasite to try to escape out of there.
Think about it that also when you lower temperature in mice, I'm not so sure if it really represents lowering temperature in humans, correct? Because mice, when they have whatever is called fever for us, in mice is going lower, correct? Could it be because in endemic areas, even Sylvia's work shows that the vast majority of the people and children, they all have parasites in circulation. Very low numbers, they almost don't replicate. They don't replicate. they are not removed by its immune system. They don't seem to go anywhere. We don't know exactly what is
But if
happening
some
there.
of them do fever, do they get immunized?
But they will only do fever probably if they have, could be that fever is the signal, is the trigger.
Second.
You already have mosquitoes here, you already can transmit. Could be. I'm just, this is even more speculation than the other, okay? So,
Okay, thank you very much, Maria.
thank you.
And we can all continue discussion if you want.